Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia. It's my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across the diverse themes in biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multiomics for guiding personalized medicine, where each week we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and hurdles associated with the advanced genomic, transcriptomic, metabolomic, and other omic methods, along with a wide variety of data analytic opportunities in data science, deep learning, AI, and spatial modeling. We are pleased to announce that today is the last day of our official application period for our Biomedical Data Science Innovation Lab. We do plan to extend this for a couple more weeks just in response to some of the activities which have happened out of Washington, D.C. this year, which I think you'll agree were a little bit distracting. And we want to make sure that people have had ample opportunity to have the time to submit their materials for us. So we're very eager to receive applications from folks working at the junction of multi-omic analysis, computational biology, machine and deep learning, as well as other methodologies used in spatial multi-omics for guiding personalized medicine. Our participants selected for this Biomedical Data Science Innovation Lab program utilize the content from seminar presentations like the one we're going to see today as essential material in our grant development workshop, which will be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We're super excited about our program where junior faculty, biomedical researchers, and computational experts will come together to develop joint projects on the topic of quantitative approaches and applications for spatial multi-omic data analytics. For today's seminar, I am delighted to welcome our speaker, Professor Jingyi Jessica Lee from the University of California, Los Angeles, where she is one of our BDSIL mentors this year. So we're very excited about having her. She is a professor of statistics and data science. She's also affiliated with biostatistics, computational medicine, human genetics, and leads a research group called the Junction of Statistics and Biology at UCLA in Westwood. Dr. Lee focuses on developing interpretable statistical methods for biomedical data, particularly single-cell and spatial transcriptomic data, with a strong emphasis on statistical rigor through the use of synthetic controls. She is a recipient of multiple grant awards, including an NSF Career Award, Sloan Research Fellowship, an ISCB Overton Prize, and the COPS Emerging Leader Award for her contributions being recognized across the field of computational biology and statistics. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Professor Lee's lecture today is entitled Permutation Enhances the Rigor of Genomics Data Analysis. Professor Lee will remind us that ensuring reliability in genomics data analysis is a crucial component of being able to make useful inferences. And her presentation will introduce two statistical methods, if I get this right, scDEED and mcRigor, which employ a permutation-based techniques to improve the reliability in single-cell data analyses with a focus on data visualization and sparsity reduction. The core message that we will hear from her is to use permutation as a flexible, assumption-lean technique to describe and establish an approximate numerical background and reference for detecting dubious findings in data analysis of transcriptomic data. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching us on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are strongly encouraged to submit any questions they may have via their chat feature in their YouTube sessions. I will be synthesizing all of these questions and I'll ask them on your behalf in the last 10 minutes or so of Jessica's lecture. So with that, Jessica, thank you so much for joining us. We're really looking forward to your presentation.
Thank you very much, Professor Van Horn, for your very nice introduction and for the invitation. So I just want to echo for the invitation to apply for this BDSI, Biomedical Data Science Innovations Lab, this coming summer, and I'll be one of the mentors. So you're very welcome to attend if you're a junior faculty interested in spatial multiomics. So I'll begin my talk by introducing, I think, this very interesting cartoon I found on social media after the Nobel Prize in Physics was awarded this year. So there were a lot of discussions about whether this award is a physics award. And then this can be rooted in the definition of physics. What is physics? So I found this media picture to be very interesting because it defines physics in two dimensions, the object to study and the method to use. So for each dimension, it goes from being very fundamental to being very liberal. In other words, being fundamental, it is the classical object, classical method we use to define the discipline in the beginning. But being liberal means that we are going broader. So for example, neural network is not a physical object in reality. Is it an algorithm, right? We arbitrarily, we manually create. And also, the method we use can also be going from the theories to be fundamental or anything that we can study the object. So in this case, the neural network is in the liberal side of both dimensions, not traditional object, not traditional method. So this comparison made me think about genomics, whether it is a liberal discipline or not. And my answer is yes. I think for multiple reasons. First of all, it is not a traditional discipline, it's interdisciplinary from the beginning. And it usually generates data before having a scientific hypothesis. So therefore, it's data-driven. And also, as the field is very popular, we have a rapid evolution of computational methods. Therefore, for anyone who do genomic data analysis, they could have flexible analytical approaches. So just using single-cell genomics data analysis as one example, we have this sharp rise in the number of methods available, and this will continue rising. So this made me wonder, my other field, statistics, is it still relevant? Especially in the current era of method and data analysis liberalism, and also the rising AI technologies, do we still need these traditional statistical approaches for our research, for our data analysis? To answer this question, I think back in history. What is statistics? Why was it a discipline in the first place? So looking back, I think statistics is a discipline that ensures rigor in data analysis. Otherwise, if we just have data, it's a set of numbers, then there are many ways, right? Numerous ways for us to operate on the data and get something out of it. But what is the guarantee? So if we just talk about this famous two-sample t-test, it was started in a brewery in Dublin, Ireland, and the author or the developer of the two-sample t-test was so modest that he called himself the pseudonym student in the paper. But the question is very practical. That is, suppose that you are in the brewery, you have a new ways to brew your beer. Would you say that the quality of the beer is improved with the new way compared to the old way? How do we answer this question? So people will collect some measurements about data quality for the beer brewed in the old way and the beer brewed in the new way, right? So we'll have two sets of numbers to compare. Even though we don't know statistics, our intuition would be that we should just compute an average of the numbers in each set, and then we compare the two averages to see whether the average of the new way, second set, is higher, larger than the average of the first way. However, how large is the difference large enough so we can be sure that there is indeed improvement in the way we brew beers? So the two-sample t-test was designed to ask this question. That is, we're comparing the difference we observe between the two averages to something we can expect under random chance. So following this, I will also say that this is a very nicely written paper that summarized the most important statistical ideas in the past 50 years. So even though under each idea, we have many methods developed for different purposes, but the essence is in the ideas. So in particular, one idea is what I will talk about today, permutation. So permutation was put under this big idea as bootstrapping and simulation-based inference. In other words, we're using simulation to help us understand our data. And permutation is a way to simulate. Basically, what we try to do is to resample our data to break some possible relationships between variables. So therefore, we know in the permuted data, the relationships among the variables are gone. And we can think, okay, there's no relationship among the variables of my interest. So I can use the permuted data as a negative control. That's the general idea. So I'll talk about two settings. First, supervised learning setting briefly, using bulk RNA-seq data as just a teaser example. So in supervised learning setting, each sample, so in our transcriptomics, it could be a bulk sample or it could be a cell. And the samples depend on your experiment, how many samples you collect, and different experiments will have different sets of samples. But the features are the variables that are consistent or comparable across experiments. So in our case, for transcriptomics, the features are genes, because no matter what samples we collect, if it's the same species like human, we should have the same set of genes. So for supervised learning, usually every sample has a label besides its features. So the question is to study the relationship between the features and the label. So in bulk RNA-seq here, I will use the example where the labels are sample condition labels, like treated versus untreated or healthy versus diseased. So to compare the two conditions for the features. And later, the theme or the major part of my talk will be unsupervised learning. Here, the samples will be cells and the features will be genes. And the question we want to ask is two things. Dr. Van Horn introduced in the beginning. We will look at how we can visualize the cells by projecting the features into a two-dimensional space and can we say the visualization is trustworthy or not? And the second question is, can we reliably aggregate our cells into small clusters? So we call those metacells. Then we use the small clusters instead of the original cells for better data quality. That's the hope. So I'll first talk about the bulk RNA-seq example where we have sample condition labels. So this teaser example was based on a publication we published almost 3 years ago. So in this, I would say a surprising work, a side project, I collaborated with Dr. Wei Li at UC Irvine, who's a computational biologist, and his former postdoc Yu Mei, who's now a faculty herself in China, and my former student and postdoc Xinzhou Shang, who's now a faculty member at Oregon State. So in this work, we found by coincidence that if we just apply popular differential expression methods to compare two conditions of bulk RNA-seq data on human population samples, in other words, each sample is from one individual, not from experimental replicates. So we could have exaggerated false positives much more than expected. So this is the setting where we first noticed this issue. And on this data set, here we have 51 patients pre-immunotherapy anti-PD-1 treatment and 58 patients on treatment. And the samples are their blood samples. We do bulk RNA-seq. All right, so the Cell paper reported the data set. And what we did is if we apply a popular DE method or any DE method to get DE genes between the two conditions. Our first observation was that two popular methods people will for sure learn when they learn RNA-seq data analysis, DE-seq2 and EDGE-R, give us different numbers of DE genes. And the number reported by EDGE-R, the red dot, is almost twice as that of DE-seq2. So we've tried to understand the differences in the results. But one thing that drew our attention was that here the sample sizes are much bigger than the scenario when both methods were developed for more than 10 years ago. Because at that time, RNA-seq was a new technology, the cost was high, and also for many wet labs that generated RNA-seq data from their biological conditions like wild type or mutation, they usually only generated 3 samples or 3 replicates per condition. So with such small sample sizes, 3 versus 3, The hope is that we can still identify the genes, so we can have some discoveries for downstream analysis. So to compensate the small sample size, both DESeq2 and HR made one important assumption. That is, every gene's read count in one condition follow a negative binomial distribution. And then to identify DE genes, they are basically comparing the two negative binomial distributions mean parameters, and a DE gene is called as those whose mean parameters are different between the two conditions. That's the basic idea. But they tackle or they handle replicates. Here, it's maybe a bit too much to assume individuals as replicates because we know there's much greater heterogeneity among individuals. And whether negative binomial is still a reasonable assumption, that is the question. So what we did is that to leverage the large sample size by just doing very simple random permutation. That is, we keep the sizes as 51, 58, but we randomly allocate the total of 109 individuals to the two groups with the sizes 51, 58. And the two groups have random labels, right? So we still call them as pre-treatment but on-treatment, but the individuals were randomly assigned to these two groups. So this is the negative control data we created. And we believe that it doesn't make much sense to find reliable DE genes from these permuted data. So what we did is that after each permutation, we run each method again. And then as we can do the permutation for thousands of times, 1,000 times, we will get a distribution for the number of DE genes reported by each method. And this bar and the error bar reports the mean and the standard deviation of that distribution. So what is most surprising is that on many permuted data, we could get more DE genes than what we did from the original data. And also, if we look at the DE genes found from the original data, many of them were also found from permuted data. So this raised a very big question mark, why this could happen. So we look at the negative binomial assumption I just introduced, and we see that using this technique called quantile plot to compare the distribution, like HR fitted to the data points themselves. So we can see that here on the QQ plot, there are some clear outliers that don't follow negative binomial distribution well, because under the fitted negative binomial distribution, we should expect the existence of so many outliers. And we see that for both conditions on this data set, and similar phenomenon for DESeq2. So that was the major reason we thought that drove this surprising result. That is the negative binomial assumption, which we assume for experimental replicates, and which is something we have to assume to get good power with small sample sizes 3 and 3. Because with 3 and 3, you will only have 3 dots here and 3 dots here. So that won't give you enough signal from data to tell you whether negative binomial is reasonable or not. So basically the negative binomial assumption is more like theoretically. It's what we're like theoretically driven. We use the theory, we understand the data generation to make this assumption. But with this data set, not replicates and much larger sample size, then we can see negative binomial doesn't fit very well. So I think this is the major message I hope to provide as a teaser. That is, we acknowledge that this is not a complete benchmark study, and we're not trying to advocate to say, oh, even though we found Wilcoxon here as a robust method, but we're not saying it's the best method compared to alternatives because we didn't do a complete benchmark. But the message here is permutation is a very useful and simple technique that people can leverage for their sanity checks. Therefore, when you use a popular method, you can think about how to do a sanity check, then you can be more sure of the results you found. So that's the teaser example. Now I'll move on to the major part, single-cell RNA-seq. So I'll talk about two things. One, visualization. One, how to aggregate single cells into metacells. So for visualization, t-SNE and UMAP are most widely used for visualizing cells. So the goal is to look at each dot in our 2D plot as a cell, so we can make some interpretation of the cell-cell relationships or cell type relationships, or whether the cells follow a continuous trajectory. t-SNE was developed in 2008 and became popular for single-cell data analysis. But t-SNE has a key parameter, hyperparameter called perplexity, which defines the size of neighborhood for each cell, because what t-SNE aims to achieve is to preserve local proximity of cells or data points so that you can see cluster patterns clearly when you look at single cells. And these clusters will tell you, oh, they might be cell types. So people want to see those clear clusters, which PCA unfortunately usually don't show, because PCA try to preserve the global variance. So therefore, you may see your clusters adjacent to each other in PCA, but t-SNE will show you distinct clusters. However, there's no free lunch because the perplexity can be tuned and it can vary. So when it's very small or when it's very big, you can see very different patterns. And this is a toy example in which the ground truth, the data is just two-dimensional. So you can see this as the true image, but then you can see for some perplexity, maybe the t-SNE visualization resembles the truth, but for others, you may be very far off. So if we want to use the t-SNE or UMAP plot to interpret some relationships among cell types using the clusters distances, we should be careful. And this warning message was raised in this recent commentary article on Nature Methods. The take-home message is that sometimes these methods take a second thought, how much you can trust the visualization. So our work at scDEED, which was published in Nature Communications last year, try to address this problem by providing an add-on to t-SNE and UMAP. And our idea is very simple. We try to answer the question that is whether a cell's embedding is dubious or trustworthy. How do we define this, right? We try to examine the cell's neighbors before and after embedding. So we hope that the two sets of neighbors are similar, and that will give us some trustworthiness of the embedding of the cell. This is a collaboration with my collaborator, who is a statistician, Dr. Lucy Xia in Hong Kong, and also my PhD student, Christy at UCLA. So the intuition here is for each cell, we look at two sets of neighbors. One is in the pre-embedding space, one is in the 2D space. So how do we define the pre-embedding space? A clarification here is that in single-cell data analysis, the data people input into t-SNE or UMAP are not the original gene expression data, where the dimensions are the genes, right? And thousands of dimensions. That's too high dimensional. And why we don't input them directly into t-SNE or UMAP? The reason is that both methods will rely on preserving local Euclidean distances of the local neighbors. So Euclidean distance will be used, but it is well known that Euclidean distance is not trustworthy or meaningful in very high dimensions. So therefore, the first step is typically PCA or similar techniques to reduce the number of dimensions from 1,000 to 50, 20 to 50, a much smaller number. And then every cell will be embedded or will be encoded by the PCs, 50 PCs, and then these PCs or these cells in PC space will be input into t-SNE or UMAP. So therefore, here the pre-embedded neighbors are based on the PC space, and then we use the Euclidean distances among cells in the PC space to define the pre-embedded neighbors. For the 2D space, we'll just use the 2D Euclidean distances. That's what we visualize when we see the plot to define the neighbors. What is the neighborhood size? This is the key thing. We define by default half of all cells as the neighbors. The reason of defining such a large neighborhood is to preserve mid-range distances, especially the distances among cell types, right? Not just the local neighborhood. Because if you define the neighborhood size to be very small, say 10%, then we find that, as we expected, t-SNE and UMAP can do very well across their perplexity or other hyperparameters. The reason is they are designed to preserve local neighborhoods. But if we define the neighborhood size to be bigger, then we are looking at more mid-range or longer range relationships. So once we have the two sets of ordered neighbor cells, so from the closest to the farthest, like half of all cells in each space, we will take the set and then we will record each neighbor cell's distance to the cell in the 2D space. So in other words, even though the two sets have their respective different spaces, but when I try to construct this distance vector whose length is equal to half of all cells, I would use 2D Euclidean distances as the values. So I'm just filling the values based on the neighbors, who they are, and their corresponding order. So the reason I want to do this is because when I want to check the consistency of the two sets of neighbors, I want to use their Euclidean distances we see in the 2D space, because that's how we interpret data. And then my hope is to give a score, like we call it reliability score, to measure the similarity between the two vectors. How do we define it? We use the Pearson correlation. The reason is that Pearson correlation is sensitive to the magnitude of the values. And that's what we care about. We're not just caring about the rankings of the distances of the two sets, but we care about whether there are some larger distance values, and that should be reflected. So that's why we use the Pearson correlation. And in this case, if the two sets of neighbors and their orders completely agree with each other, then this Pearson correlation will be equal to 1. So that is the perfect score we can give. It ranges from -1 to +1. The second example is a bad embedding, or we call it dubious embedding. So in which case, the pre-embedding neighbors, if we look at them in the 2D space, they don't seem to be reasonable, and they're very different from the 2D neighbors here. So in this case, the reliability score will be low. But here the question is, if we want to just set a threshold for us to dichotomize our interpretation of embeddings, to call some as trustworthy, some as dubious, some as uncertain, then we need thresholds. How do we define the thresholds on our reliability scores? We use permutation. Why it can help us? The reason is that we need to create a negative control case in which a cell's neighbors are just a random subset of all cells, like half of all cells selected randomly after embedding. So this is the case where the embedding does not preserve any neighbors reliably, right? Because it's just random sample. So how can we create such a case? Then permutation can help us. So in other words, the permutation we're doing here is that we independently permute or shuffle the values of each gene across all cells. So what do we preserve and what do we disrupt in doing so? First of all, we preserve each gene's distribution. The reason is that the gene's values remain the same values. It's just that their order is shuffled. So the distribution for this gene alone is the same. But what is not preserved, first of all, as we do the permutation independently for all genes, gene-gene correlations are gone. And also, as we do this independent shuffling, you see the values are randomly assigned to these cells for each gene. So the cells are no longer the original cells. I call them permuted cells because they are new entities that we don't measure by experiment. But these permuted cells have something very interesting. That is, each cell can be represented as another copy of another permuted cell. So they are called as identically and independently distributed, IID in statistical language, after such permutation. So basically, they can be treated as equivalent to each other. So there's no more meaningful cell-cell relationships among these permuted cells. So therefore, if we visualize those permuted cells, who are the neighbors of each other, it's just a random thing. And this is something we hope to say. So with permuted data, if we apply our reliability score calculation, then the score will just reflect the case where the cell's neighbors or the pre-embedding neighbors and 2D embedding neighbors are just random. Though they don't really have any meaningful consistency there. And that reflects the negative control case we can think of. So in other words, the magic, or I would say not magic, the interesting thing here, which is also something people tend to criticize for permutation, that is, it's too computationally intensive. But we don't have this issue here because we just need one round of permutation. Because after one round of permutation, we get n permuted cells, and each permuted cell will give me one reliability score under the null. The reason we can do this is because our score is a statistic. We compute for each cell. It's not a statistic for the whole data set. Otherwise, we do need to do many rounds of permutation to get many copies of this data set. However, here we're looking at the per cell statistic. So here, once we do one round of permutation, we can already get n values of the null score, null statistic, and that can give us a distribution. This is the basis for our threshold decision or determining. That is, if we believe a real cell or original cell's embedding is trustworthy, then its reliability score must be much better than most of the null scores. So that's why we determine this threshold, the right tail, for our trustworthy calling. On the other end, if a real cell score is even lower than the majority of the null scores, then that's really bad, and then we can call it dubious. And for the middle part, how much do we set in the middle? We follow the convention in our default to set the middle as 95% probability, which means that the two tails add up to 5% probability. But this doesn't mean that for the real cells, we can only classify 5% of them. Not the case, because here the null distribution is based on the permuted cell, the null cases, not the real cells. So in other words, we will see how many of our real cells fall into the two tails. And based on empirical evidence, the good thing is that the majority of embeddings for t-SNE and UMAP of the data sets we examined are in the trustworthy region. A small minority in the dubious region, that's worthy our attention, and very few in the middle. So this is one example of this Hydra single-cell RNA-seq dataset published in Science in 2019. It's the first single-cell RNA-seq dataset of this species in the ocean. So this is the original t-SNE plot using a default perplexity of t-SNE. We can see many small clusters representing hypothetical cell types, or some of them have the same cell type label but different numbers like 2, 5, because they are in different clusters, but they cannot be distinguished based on biological knowledge. So that's why the authors give them just different numbers. So one thing we noticed very interestingly, that is running scDEED on this setting, on this visualization, we find the dubious embeddings in small clusters. So this may suggest that these embeddings, these small clusters locations are not trustworthy, or we cannot make too much story out of where they are and their distances to other cells. And one other functionality of scDEED is to optimize the perplexity by minimizing the number of dubious embeddings. Because here, at the default perplexity, we can detect dubious embeddings and we can get a number, we can count them. And if we change the perplexity parameter to a different value, we get a different visualization, then we can count the number of dubious embeddings. So we can do that until we think, oh, the number of dubious embeddings as we increase perplexity reaches a plateau. It decreases and it no longer decreases much, like an elbow thing. Then we can stop there and report the perplexity. Or we can minimize the number of dubious embeddings by doing a grid search across different perplexity values. So you see, at the perplexity we choose with scDEED, the plot, the visualization looks very different from the original one. In particular, we can focus on these clusters, the orange ones, stem cells, and these three clusters surrounding it, it's called neuron ectoderm cells. So with our visualization, you see that the three small clusters are now unified down here. And this is consistent with their labels. They are the same cell type label despite their different numbers for different cluster index. And also, we see that this unified cluster is very far away from stem cells. So unlike here, they're very close to each other. And you see these new distances we see are more consistent with the data, the gene expression heat map. We do see the stem cells are very different from these clusters, and these clusters themselves are much more similar. So this gives me one kind of, comparison to show whether we think the visualization is more reliable, and we go back to gene expression data. Okay, and another thing, we have multiple results in our paper, but one thing I want to just say is that using our optimization for t-SNE and also similarly for UMAP, it's just that UMAP has two parameters, minimum distance and number of neighbors. We have to optimize jointly by doing a 2D grid search. And we can see that with optimization, something pleasant to see is that the consistency between TC and UMAP is improved. That is, for this very famous PBMC blood data set, and they were measured by different technologies of single-cell RNA-seq, and we see that for these 4 major cell types, so the relationships or distances of the cell types became more consistent with our optimized visualization between TC and UMAP. So the thing is that whether the cell types follow a counterclockwise order or clockwise order, the orders are consistent between the two techniques, TC and UMAP now, but they were not as consistent before our optimization. So this also says that basically, the difference of TSD and UMAP depends on many things, including initialization and also how to set hyperparameters. Okay, that concludes my part 1 about the visualization. Now I'll move on to part 2 regarding Metacell. So what is a Metacell and why do we need Metacell? So Metacell was introduced as a technique which is like a very fine-grained clustering to reduce the sparsity in single-cell RNA-seq data and also other single-cell omics data. That is, you have just too many zeros than your expectation. And many genes that should be expressed were not detected because of the low sensitivity of the technology at the per cell level. So the sparsity is the big thing. And what Metacell helps to do is that by finding similar cells in small clusters from the gene expression profile or other omics profile, then we just define an average for each metacell and then use the average as the metacell profile. And then we just use metacell profiles instead of single cell profiles for downstream analysis. And besides the sparsity reduction, another advantage is that the metacell number is much smaller than single cell number. So we may also gain some computational efficiency for downstream analysis. But from a statistical point of view, we are altering the data. And we cannot just alter the data in any way, right? Any way what we want. We need some justification. So my question is, what is a reasonable definition for aggregating cells into metacells? So in the field, there are 4 Metacell papers, and the first one, Metacell, introduced the concept and the term, and there's a recent one, C cells. So they have been used, and also there are applications of the Metacell concept in these data analysis and generation papers in which people do Metacell construction in their own way. They may not using these methods. So in other words, there lacks a consensus on Metacell definition. And to address this question, to address this issue, we propose a method called mcRigor. So MC stands for metacell, and we hope to answer 3 questions. First, how to define a metacell. Second, how to detect dubious metacells that don't agree with our definition. Third, how to optimize metacell partitioning based on our pruning by removing metacells, dubious metacells. And this work was led by my postdoc, Panu, in my lab. And this work has been accepted by the RACON conference, the leading conference of computational biology and bioinformatics, and it's currently under revision at Nature Communications. So we will first go back to the metacell definition in the first paper, metacell algorithm. So this is a quote. It says, a metacell is a homogeneous collection of single cell profiles that could have been resampled from the same original cell. We translate this definition into a statistical language. That is, the variation within the metacell is attributed exclusively to a measurement error in the sequencing experiment. It is not due to biological differences of those single cells. So how can we formalize this definition? We thought about this very insightful paper from Dr. Matthew Stephens at UChicago, and which proposes a 2-layer model for single cell data, an expression model as the bottom layer to model the distribution of true gene expression levels across single cells. Suppose that we know the truth. And the measurement model is about the second layer, which is the distribution of our observed counts, the UMI counts or read counts. It's typically UMI counts nowadays with the technologies, given the true expression levels. So observed counts are our data, and true expression levels are the hidden truth. So this 2-layer can help us clarify what we mean by the variation due to measurement error. So specifically, we give cell I this index, and we give feature J or gene J index J. So cell I, gene J. So expression model is about cell I's true expression, but we must bear in mind that the sequencing experiment can never give us absolute expression. It only provides relative expression. So therefore, the true expression level is a true proportion vector, expression proportion vector. So lambda I is p-dimensional, corresponding to the p genes' true proportions in cell I. And we assume that for each cell I, the true proportion vector follows some expression model conditional on cell I's covariate. And by covariate, I mean the cell I's cell type or cell I's cell state. So those things that can determine its expression, gene expression levels. So I'll get this lambda I vector. It's p-dimensional. Lambda ij is the j gene's proportion, expression proportion in cell I. And given this true proportion vector, the measurement model, the sequencing step, can be reasonably assumed as something like a multinomial sampling. So given cell I's total reads, suppose we assume this yi plus is the total, we assign it into separate genes based on the gene's proportion. Okay, so the yij is the gene's j's count in cell I. So if we think this is the measurement model, if this is reasonable, then we can come up with a statistical definition for Metacell. That is, it is a group of single cells that share the same lambda, share the same true expression proportion, but they differ in their gene expression count for those single cells because of the measurement model. So if a metacell satisfies this definition, we call it trustworthy. Otherwise, we call it dubious. So this becomes a statistical problem. So first of all, let me explain why dubious metacells are serious issues. So this is the Metacell review paper that's published already. So basically, we hope that in each metacell, the single cells included have the same cell state. But if in this cartoon, like a metacell includes cells of two different types, and if we're using the metacells instead of single cells to do some gene co-expression analysis to say whether there is any positive, negative correlation between two genes. This is what we get from single cell. Because single cell has a lot of zeros, so many cells have gene A close to 0, many cells have gene B close to 0. So we don't see a very clear relationship or co-expression between the two genes. But if we use metacells, and there's a dubious metacell that combines two cell types, then it can give you this false positive correlation if you do gene correlation calculation based on metacells. So that's the problem. So our mcRigor method will provide a statistical criterion to identify dubious metacells that consist of single cells from different cell states. And besides that, we can help optimize an existing metacell algorithm by optimizing its hyperparameter called granularity level, gamma, which is the average number of single cells per metacell. And we hope to choose this hyperparameter in a data-driven way. So you see the high-level idea resemble that of scDEED. We're trying to detect dubious things, and we're trying to help an algorithm optimize. So we're providing an add-on to an existing algorithm instead of replacing the algorithm totally. So here's our intuition. That is, if we believe that within a trustworthy metacells, the single cells only differ by the measurement model, then the genes or features should be approximately uncorrelated. We must acknowledge that, so the multinomial distribution in the metacell, in the multinomials, in the measurement model, this multinomial distribution will induce some correlations among the genes, but this correlation should be minimal. And this is something we hope to capture by generating our negative control by permutation. So this intuition will give us something like this. In a trustworthy metacell, if we plot this gene-gene correlation matrix, we shouldn't see some very clear block pattern to indicate that a block of genes are correlated. So here you see the diagonal means the gene's correlation to itself, it should be 1. But off diagonal, we see only very spurious things that they don't have clear patterns. While if it's a dubious metacell, includes cells of different states, then we may expect that the different states will drive strong gene-gene correlations, and these genes will be in the block if they function in the pathway, for example. So our strategy here is to quantify the per metacell gene-gene correlation matrix by a statistic, and we call that mcDIV, stands for metacell deviance. And we also hope to preserve cell library sizes for each cell because that would induce gene-gene correlations at a in the minimal way in the measurement model. So we want to preserve that. So the first question I want to ask is, can we just use within gene permutation as in scDEED? So here, genes will become uncorrelated, which is something we expect for trustworthy metacell, but the cell library sizes are gone because the permuted cells don't correspond to original cells. There's no one-to-one correspondence. So what is our strategy? So my postdoc Pan came up with this very smart idea. We call it double permutation. It has two rounds of permutation. So first of all, we did do the within gene permutation in the same way as scDEED. So basically, this uncorrelated case will give me some normalization factor for the gene-gene correlation matrix. So I'll introduce the formula next slide. So this way preserves the genes because each gene is still the original gene, although we remove gene-gene correlations. But we also remove cell library sizes. Then how can we compensate that? So in the bottom, we'll first do within cell permutation. That is, we shuffle all genes values within each cell. So the good thing here is that the cells are still the original cells library sizes because we just shuffle the values, but the library size stays. And these cells correspond to the original cells. So in this case, we can compute the permuted genes, these genes no longer original genes. For permuted genes, we can compute their correlations, but they are like just due to the, the random thing, right? Because these genes shouldn't have any correspondence anymore, and they should represent the uncorrelated case. However, when we normalize for these genes on the top, right, because this will give us a purely uncorrelated case since we shuffle each gene independently. That's purely uncorrelated. And here, for permuted genes, even though we think by shuffling the values, they don't mean something meaningful, and they should represent the case where the metacell is pure. However, we need its own normalization factor because the permuted genes are not the same as original genes. So that's why following this, we do another round of within gene permutation within each permuted gene. Then this final double permuted matrix can create a normalization factor for this matrix, for this gene-gene correlation matrix. So together, the bottom row will give me a null value for mcDIV. So this is what we did exactly. So we define mcDIV as a ratio, which is the Frobenius norm of the difference between the gene-gene correlation matrix within each metacell and identity matrix. Because identity matrix is the ideal uncorrelated feature-feature gene-gene correlation matrix, right? So we calculate the deviance from that. And we normalize this Frobenius norm by using the gene-gene correlation matrix from within feature permutation. So that's what we did with the over here, top right here. That gave us a normalization factor here. And we need this because also we need to account for each feature's distribution. Second, we do that with the within cell permuted case, the bottom negative control. So we do the permutation within each cell, and then this will give me the data, permuted data, and then we get the null mcDIV based on the further within gene permutation given the permuted genes. So we basically do the same ratio, except that the calculation's done after we do the within cell permutation. So here the cell library sizes are preserved, but the genes are permuted. So from the null cases, again, we can get one null value per metacell. So we also only need one round of permutation. We can get this relationship, the null values we realized that they actually have some dependency on the metacell size. That is how many single cells are in each metacell. So therefore, we need to derive size-dependent thresholds for the MCDIV, DIV null scores. So this will give us, okay, given the MCDIV, if the score is too high, given the metacell size, then we flag it as dubious because we know the larger the score here, the worse, because it means that the feature-feature gene correlation matrix is more deviated from identity. So that's how we try to distinguish or discern each real metacell before permutation, is it dubious or trustworthy. So due to the time, I'll be quick to go through the rest of my results. So first of all, we justify using simulation that double permutation is needed. Within gene permutation is not enough because we will flag too many metacells as dubious, even though in the simulation we know the ground truth. But we only use double permutation. It's a better negative control than the within gene permutation. Then the identification accuracy is much better. And also, we use this very interesting data set in which there are so-called multiplets. That is, you can have multiple cell barcodes enclosed with one single cell in a droplet. So therefore, even though these barcodes are capturing the same cell, but if you look at the data, if you don't know they are the multiplet, you will think they're different cells. So therefore, these barcodes will represent the measurement error, and we can consider them to be the single cells that should have been aggregated in the trustworthy metacell. And indeed, we test mcRigor on this data set and found that these multiplets are all flagged as trustworthy. And this is a very interesting result from the supercell method paper for metacell construction. Using single cells or all metacells, they compare the gene-gene correlations in healthy patients' blood data, single cell data, and COVID-19 patients. So we focus on one gene module called adaptive immune response gene module. And we see that using single cell data, because of sparsity, we don't see many strong correlations within the module. Using all metacells, we see much stronger correlation, but the two correlation blocks are similar between healthy and COVID-19 patients. However, using our approach to filter out dubious metacells, we see a much bigger difference. So in healthy patients, not so much correlations, but in COVID-19 patients, much more correlation among these adaptive immune response genes. And that is consistent with our biology. We know during the COVID infection, these genes will be active. If we go back to the data, we see that the single cells, for like some correlation, the single cell data don't show strong correlation like in this case, but in this case, single cell data does show something, right? If you look at the non-zeros. But if you use all metacells, then the blue ones are what we flag as dubious, you will see that they will drive strong but positive and false positive correlation as something we can avoid by removing those dubious metacells. And this is a single cell multi-omics analysis. Basically, we have both a tag-seq and RNA-seq for chromatin accessibility and gene expression. The hope is to identify enhancer gene correlations. So using single cells and focusing on the gene GATA2, we don't see many enhancers associated with the gene. Using all metacells, we have many more, and this is the result from the C cells paper. Using our way to filter out dubious metacells, we removed the red edge, but we added the blue edge. So we try to convince ourselves that the removal and addition are reasonable. So first of all, the edge we remove, if we look at tag-seq data, we do see very, very minimal signal there. So it doesn't convince us that there should be an enhancer. But if we look at this added blue edge, we look at the data, and there is some signal, and it is a validated enhancer in the literature. So we look at the single cell data for further confirmation. We do see that for the blue edge, if we look at the non-zeros, we do see some relationship between enhancer and gene. But for the red edge, if we look at non-zeros in single cell data, there's not so much signal. So finally, I want to say that by optimizing the gamma parameter, which is the average number of single cells per metacell, we can see that at our optimized resolution gamma for each of the three metacell algorithms, metacell, C cells, supercells, at the corresponding resolution, we can see that using the corresponding metacells at that gamma to calculate zero proportion, these proportions are more consistent with the fish data. So in this very interesting data set, people did single-cell RNA-seq and fish, and using fish as the ground truth or gold standard data is more accurate. So this confirms that, okay, if we believe the zero proportion of fish is the biological zero proportion, then using our way to optimize Metacell definition, we can better approximate the proportion of biological zeros, because we're hoping that Metacells can help us remove technical zeros. So I'll stop here by summarizing that I talk about bulk RNA-seq DE analysis as a teaser, as a supervised version. Here it's simple that we just permute sample condition labels, but later I introduced two ways to do permutation for single cell data in the unsupervised setting, where we do within gene permutation to get permuted cells without relationships. And for mcRigor, we did a more complicated double permutation, so we can preserve cell library sizes first and then start from there. So finally, I want to thank my collaborators and students and postdocs for their work and for the work they did for this talk. Thank you very much.
Oh, thank you, Jessica. That was really fantastic. Yeah, I mean, it's just really interesting for a number of reasons. And one in particular is that just t-SNE shows up everywhere. It's not just in single cell, data analytics. It's being used in all manner of things. And I do have a sneaking suspicion that it's used a little bit casually as sort of like, oh yes, this is just what you do, but people don't often ask the underlying questions about exactly how much is it changing our data. And I'm glad that you kind of talked a little bit about that. And you have your way of looking at, what you call dubious, basically dubious points within those plots. I'm kind of curious, given that it's so ubiquitous in all of these different types of things that its application is that in UMaps as well. How prevalent do you think that dubious findings are in the literature, if I might pin you down a little bit?
Very good question. I would say maybe the most serious issue is that I've heard many people, when they talk about TC and UMAP, they mistake them for clustering method. I think that is the biggest issue. They think, oh, we're doing clustering with TC and UMAP, but they are not. They are just doing visualization. So therefore, if people see some distinct clusters in TC or UMAP, they might think, oh, I found a new cell type, or this cell group is distinct from the rest. But that could be due to an artifact of the visualization. So to me, that is probably the most prevalent issue, but we haven't done anything like to do the literature to rerun their every analysis, right? That's a lot of work. But
Yeah.
yeah, but if anyone is willing to do that, I'm sure there will be something that can be found.
That
Yeah.
would be, you're absolutely right, a large amount of work. We had a question that a person had asked about, DE-seq2 and EDGE-R make assumptions about gene-specific dispersion parameters. Could that be one of the reasons why the fit is so poor in real data rather than the kind of null distribution assumption
Good
data?
question. Yeah, that's a very good question. I ignored that technical aspect. But in fact, negative binomial is a two-parameter distribution. And how well you estimate the dispersion parameter will also decide whether you have a valid test for the mean parameter difference. So it's intertwined. So you need to estimate the dispersion parameter well. And for both TC and UMAP, for both EDGE-R and DE-seq2, since they handle the small sample case by 3 versus 3, that the sample size is too small to estimate the dispersion well. So they use something called like shrinkage or empirical Bayes approach to borrow information across all genes. So then they can help each other learn the dispersion parameter. But it is also well known that doing so, you will introduce some bias in your dispersion parameter estimate. And how large is that influence on the result? That is something we didn't quantify, but it is a factor there. So it's not just negative binomial assumption itself, but if a negative binomial distribution doesn't fit well at all, then that's the more serious issue. But even if it fits well, then the dispersion parameter accuracy is a more, how to say, detailed but important factor to decide to determine the false positives. And so I know someone who did. There's a recent preprint by Tim Barry, and he's a postdoc at Harvard School of Public Health. So he had a preprint paper, very interesting, on archive 5, which is about using permutation to correct for a negative binomial regression, which is used in both Agile and DSEQ too. And this can help remove some of the false discoveries.
This is great.
Yeah.
I have one more question for you, and it's in your McDiv ratio that you
Yeah.
calculate, where you're looking at that norm of the kind of
Yeah.
correlation matrix minus
Yeah.
the identity matrix. I'm just
Yeah.
kind of curious because of course, the same thing would be
Yeah.
achievable, I think, by looking at the eigenvalues of that correlation matrix.
Mm,
And then you've got basically the ratio of a product of
mm,
eigenvalues and whether or not that isn't somehow useful for
mm.
determining underlying dimensionality, because things should cancel, and it may give you some insights into, how much of a compression you're going through when you then project it into a two-dimensional space like you're doing with t-SNE. That's just
So,
a random thought, but I thought I'd
yeah.
chuck it out there.
Thank you very much for the very interesting thought. So first of all, we didn't think about connecting this to TC and UMAP, so we're not thinking about dimension reduction here. And second, the reason we didn't want to do this eigen decompositions because we want to save time. Because we need to do this calculation per metacell. So that if we can have a statistic that's fast to compute, that's what we hope for. And right now, the majority of time is spent on this feature correlation matrix calculation, which
Mhm.
itself is p squared time. Yeah.
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 Yeah,
Yeah.
they're gonna be big. So
Exactly. Yeah. So we are definitely thinking about ways to make this faster because I think it has to be fast enough so people are is willing to use it. But the good thing here is that now if you just want to detect dubious metacells, then the step for calculating this MCDiff is very negligible compared to the metacell partitioning time. That's okay. But if you want to search for the gamma value to optimize that, then you have to do this calculation. You have to run basically metacell partitioning by an algorithm, say C cells, for each gamma, and then run mcRigor. So then the number of gamma values to examine should be the total time, right? So total time scale with that number of gamma values. So that is another thing. But still, I would say so far, the majority of time is spent on metacell partitioning itself. Another thing I want to add is the amount of methods we look at. So the 3 methods, for example. So yeah, so there is a clear trade-off between the performance and the speed. So C cells is the slowest, but it seems to be the best performing. So based on our evaluations. So therefore, I think there's still some room to find whether we can improve the computational efficiency while preserving the performance.
Oh, thank you, Jessica. Well,
Yeah.
let me thank you for your giving us a fantastic talk about the use of t-SNE and your some of the tools that you have developed to kind of like look for values which are untrustworthy versus those that might be, and how to make improvements on things. I'm sure that our Biomedical Data Science Innovation Lab attendees will want to talk to you at length about the application of this to their data. So I look forward to those conversations. Everyone, have a wonderful Friday, and we will look forward to seeing you next week. And thank you again, Jessica. We really appreciate your talk.
Thank you, Jack. Thank you so much. I really look forward to the summer as well.
Yes,
Yeah.
indeed. All
Yeah.
right.
Yeah.
Have